We can measure biodiversity at different scales. For example, species diversity within communities and genetic diversity within species. And we know that these different levels of biodiversity are linked. Threats to biodiversity that lead to loss of species can naturally also lead to the loss of the genetic diversity that was found within those species. The opposite is also true, but it's less straightforward to understand how loss of genetic diversity has negative consequences for communities and ecosystems, and how it can contribute to the loss of species. To understand the consequences of the loss of genetic biodiversity, we must consider how genetic variation is distributed among individuals in populations and the population processes that modify just how much genetic diversity is present. So remember that genetic variation underlies different features of organisms and their functions. So genetic diversity is important for populations because it allows individuals to respond to and also to adapt to their environment. Genetic variants or alleles that benefit an individual's function are more likely to be passed down to future generations, since the individuals with those variants will be able to produce more offspring. The offspring will inherit the genetic variation from their ancestors, and over time, adaptation occurs. So some of the population level processes that affect genetic biodiversity are stochastic or random, due to how genetic variation is inherited and due to the number of individuals present in the population. The number of reproductive individuals in a population is especially critical, since only these individuals are able to pass on their genetic variants to the next generation. Now, if the population is large, there's potentially a larger amount of genetic variation available for the next generation, and this is referred to as the gene pool. Demographic bottlenecks describe a situation when the size of the population is significantly reduced, which means that there are fewer individuals available to pass on their alleles, and the gene pool is smaller. In addition to a large gene pool, the number of offspring produced and surviving in the next generation are also important for sustaining genetic diversity in populations. If only a few individuals contribute a very large number of offspring to the next generation, the proportion of their genetic variants will increase, and it can skew the amount of diversity in the population. You can imagine these stochastic processes by thinking of a jar full of different colored beads. The jar represents our gene pool, and the different colored beads, the different genetic variants that are present in the population. If you remove a very large handful of beads to create the next generation, putting them in another jar, it's very likely that your handful will contain many different colored beads. Whereas if you only take a small handful, you will only sample some of the available colored beads. Now, if our jar contains only red and white beads, representing a gene pool that has originated from a few parental individuals, you will only sample red and white beads, regardless of whether you take a large handful or a small one, and you might only sample red beads. So populations that have experienced a population bottleneck will have less genetic variation, and even if they grow in size after the bottleneck, the amount of genetic variation available to the next generation will be much reduced. So this stochastic sampling of genetic variation to the next generation is called genetic drift. Human activity that constricts nature, such as habitat fragmentation or urbanization, can reduce the sizes of populations and shrink the available gene pools that lead to a loss of genetic diversity. And there have been many studies over the years on a wide variety of organisms that document just how human activity has constricted populations and consequently reduced genetic diversity within those populations. Of course, there's also some exceptions when genetic diversity is not affected by human activity. And these are particularly for populations that have adapted to live socially with humans or have adapted to live in urbanized environments. You can learn more about these different studies and about the importance of genetic variation conservation in the reading that is listed in the link. Genetic drift will affect all genes in the genome, regardless of whether these genes are important for a particular feature or important for the function of the organism. Moreover, because it's a stochastic process, we cannot predict which alleles will be lost, and even beneficial alleles can be lost by chance. Loss of variation at specific genes, for example, the genes involved in immune functions, can be particularly devastating to individuals and increase the risk of population extinction. But it's not just stochastic processes such as drift that can change the amount of genetic variation in a population. Artificial selection and breeding programs can also reduce genetic variation as a byproduct of focusing on maximizing the fitness or production of a specific trait in different organisms. For example, in agriculture, many crop cultivars have been bred for high yield from a small number of ancestors. These breeding practices lead to the production of genetically homogeneous crops with low genetic variation. Growing resilience on such crops and loss of the genetic variable crop varieties has led the Food and Agriculture Organization of the United Nations to describe this reduction in genetic variation through breeding as genetic erosion. This active reduction of genetic diversity through artificial selection has the same consequences for populations as does the stochastic reduction of genetic variation by drift. And although the crops have high yield, important genetic variation needed for defense against pests could be lost, making them vulnerable and less resilient, especially when they're grown in monoculture. So the trade-offs between yield and other functions of our crop species need to be considered very carefully. Unfortunately, these days, some efforts are made to protect multiple crop varieties. Now, since species are usually comprised of many different populations, the processes affecting genetic diversity in one population might not be the same as processes affecting genetic diversity in another. And this means that populations experiencing drift will potentially lose different alleles and become differentiated from one another. Populations that are small and isolated from each other will experience inbreeding as there are fewer and fewer mating partners available and because they share genetic variation inherited from common ancestors. Inbreeding further reduces genetic variation and can have negative consequences on organismal function. Therefore, it's important to recognize the differences among the populations and ensure that connections between them are maintained. Connections between populations allow individuals to move between them, bringing along potential different genetic variants. And successful reproduction of these newcomers in the population, or gene flow, increases the genetic variation and the population's potential to persist. So one strategy for protecting species, particularly those that are critically endangered, involves translocations, or human-assisted movement of individuals from one genetically variable population to other populations that lack diversity. Since genetic diversity can be measured in a variety of ways, researchers are working together to define the most essential biodiversity variables for different species to best describe genetic diversity within populations, how much genetic differentiation has occurred between them, and to estimate inbreeding and population sizes. Hopefully, these efforts will help increase the inclusion of genetic biodiversity in legislation and conservation practices and in biodiversity monitoring programs. For more information about these essential biodiversity variables, you can refer to the review article by Hoban and others.